Thank you so much for joining me. You are participating in the UBT-Rad project, if I'm correct.
Yeah.
Could you please introduce us this project?
Yeah, sure. So thank you again for giving us the opportunity to discuss this project. So this project deals with brain cancers. So we know that the incidence of pediatric brain tumors has been steadily increasing with time. While several established risk factors contribute to CNS tumors in children and adolescents, such as inherited disorders and exposure to high-dose radiation therapy, less is known about other potential causes, such as exposure to low-dose radiation. So in this study, we hypothesize that low-dose irradiation in children may play a pivotal role in the tumor formation, potentially interacting with genetic mutations to drive neoplastic development in the brain. Of course, additionally, we also would like to explore other complex mechanisms behind it that could influence brain tumor formation, focusing on three key areas, which are the specific brain microenvironment, of course, the immune response, and the redox balance.
Thank you. Can you describe your consortium and explain how you selected and engaged your partners, please?
Yeah, sure. So the consortium is composed of four different teams with diverse backgrounds. My lab, ISTCT lab, located in Caen, Normandy, in France, and supported by CNRS and the University of Caen, focuses its research on brain tumors, neuroscience, and radiation therapy. We have partnered in this project with a German team located at the Mainz University in Germany, led by Dr. Pierre-Olivier Frappard, which specializes in brain tumor development in the context of genetic mutations. Additionally, we have also partnered with a French team, also located at the University of Caen, headed by Professor Sayama Kakdous, whose expertise lies in the effect of low-dose radiation and reactive spaces. And lastly, finally, a Greek team led by Professor Sotiris Ekonomides from the EEAE from Greek, which is a Greek Atomic Energy Commission, and which is highly engaged in radiation protection and safety matters and training, rounds out the consortium.
Thank you. What key results do you expect from the project, and how do you foresee these outcomes impacting the EU community?
Yeah, so of course, so the goal is to understand the mechanisms underlying brain tumor susceptibility to low-dose irradiation in juvenile and young adults individuals, with a particular focus on the role of the brain microenvironment, immune response, and metabolic processes. Additionally, we also aim to provide new insight into the differential effect of low-dose irradiation on the brain versus out-of-brain irradiation, and to identify potential biomarkers that could predict the development of brain tumors. Last, we aim to identify factors that could be inhibited to disable brain tumor formation. So at the level of the European community, of course, new knowledge is needed to meet the growing challenge of brain cancer, which is increasing, as I discussed previously. While various origins were suspected, such as cellular form or external exposure to chemical compounds, no clear demonstration has been provided so far. But we know that individuals are exposed to low-dose radiation during the life, such as diagnostic or imaging setup, as living or occupational exposure. So we want to explore this low-dose exposure as a factor to induce brain cancers.
Incredible, thank you. Last question. Your project has just begun. What initial outcomes do you anticipate from this collaboration? And now that your project is underway, what early results or impacts do you expect to emerge from this collaboration?
Yeah, so the project starts in March 2025. So therefore, it is less than 1 year because it is exactly 5 months ago. So right now, we completed all documents relative to administrative issues, and we're also done with the data management plan. Right now, where we are is that for in vitro studies, we design all the sequences for the genetic knockdown in both astrocytes and neural stem cells that we would like to target. We also already prepared all agreements to launch the transgenic animals requested for the project. So right now, where we are, the German team has already thawed the transgenic mouse embryos so that we can quickly start breeding the mice in Caen. So the next step will be to have the animal ready to launch irradiation with a low dose, and in parallel, in September, we will start all in vitro studies in line with this project.
Thank you so much. I will stop recording now.
Okay.